Hello, everyone, and welcome to today's live broadcast, Cell Sorting for Multiple Applications, How to Optimize Your Sort Setup. I am Jen Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Sony Biotechnology. For more information about our sponsor, please visit their site at sonybiotechnology.com. Now let's get started. I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. I'd like to now introduce our presenters, Dr. Deena Soni, Global Marketing Manager, Sony Biotechnology, and Dr. Miro Koulnis, Senior Application Scientist, Sony Biotechnology. For a complete biography on our speakers, please visit the Biography tab at the top of your screen. Before we begin the presentation, we have a quick poll for you. Which professional position best applies to you? Dr. Soni, you may now begin your presentation.
Thank you for the introduction and welcome to today's webinar on optimizing sort setup for multiple application. My name is Deena Soni, and in today's presentation, I will be discussing key considerations for sorting, looking at the influence of sample preparation on a successful sort outcome. We will review how a user can set up a cell sorter using a streamlined approach, and what are the sort parameters that should be chosen during sort setup? In the second half of this webinar, my colleague Miro Koulnis will do a deep dive on setting up multiple applications using diverse samples, sharing with us how these can be set up seamlessly on the same instrument in a multi-user lab. Let's get started. Optimal sample prep is critical to successful cell sorting. As the cellular sample becomes more complex, either in terms of its heterogeneity or geometry and size, sorting them easily becomes challenging. A good sample prep can be obtained by keeping three things in mind. One, the cells should be maintained as a single cell suspension. Two, the sample concentration should be kept at recommended levels. And three, at all times, the single cells should be maintained in suspension by agitation and cooling. So here are some of the recommended approaches to generating a single cell suspension. Number one, harvest the cells using trypsin, acutase, or other enzymatic methods. Inactivate these enzymes using serum-free media, such as EDTA buffer, and then wash the cells in calcium and magnesium-free buffer to reduce clumping. Finally, add DNase to remove any free-floating DNA and keep the final sample prep on ice prior to sorting. Once a single cell suspension is obtained, the cell concentration should be adjusted depending on the type of cells that are used. At the table listed below, different types of cells and their recommended concentrations are shown. Typically, regardless of the cell size, if the sample is sticky, either due to the presence of debris or DNA, or simply because of the nature of the source from which it was derived, then such a sample should be run more dilute. In the last step, in optimizing the sample prep, keeping the cells in a single cell suspension is important. This is done by filtering them and keeping them keeping them cold both before the sort and then throughout the sorting process. Now let's look at the steps of setting up a cell sorter. The first step of sorter setup is, of course, selecting the correct nozzle. For using the correct nozzle for sorting your cells, choose an orifice that is at least 5 times the size of your cell. Smaller nozzles generate more droplets and are ideal for fast sorting of common cell types like yeast, lymphocytes, and splenocytes. As the nozzle size increases, it produces lesser number of droplets per second, but on the other hand, it provides a larger drop volume. This in turn could improve sorting of cells that are not only large, like the CHO cells, but also cells that have irregular geometry, like the neurons. Once you have selected the nozzle, the instrument must be calibrated for detecting and sorting cells. In case of the Sony sorters, it is a completely automated process where the flow cell or the chip is aligned to the lasers to ensure optimal signal on a daily basis. Next, the laser delay is determined, followed by which droplet calibration is done to determine the break-off point. In the last steps, the deflection of the side stream is optimized for 2- and 4-way sorting, and lastly, the sort delay is calculated. So let's review all the steps of setting up a system for sorting. A systematic and streamlined approach to sort setup is possible when using an automated cell sorter like the Sony MA900. On this system, the microfluidics chip represents both the flow cell and the nozzle and is available in different sizes to support sorting of different cell types. The sorting chip is a user-exchangeable consumable and includes distinct ports through which sheath and sample are injected for the cell sorting process. Also, vacuum can be drawn on the chip to eliminate any trapped air that may occur during the sorter operation. And lastly, at the bottom of the chip, there is an integrated nozzle of a defined orifice size of 70, 100, or 130 micron diameter. Setting up the MA900 can be done in 2 basic steps. In step 1, the user scans the QR code on the microfluidics chip and loads it on the system. This QR code automatically inputs baseline settings required for that chip size. Once the chip is loaded in the system, the user can then perform step 2, which is auto-calibration. In this step, the user loads auto-setup beats, and then the system takes over, and all the steps of system setup that we discussed before, which include chip alignment, laser delay, break-off optimization, side stream adjustment, and drop delay, are all executed automatically, and the system is ready for setup in less than 30 minutes. In the next steps, the different parameters required to set up a sorting experiment are selected as shown in this image. Sort setup begins with creating an experiment from a blank template or by using an existing template. A template brings in different measurement settings, including a choice of lasers and fluorescence and scatter parameters that are required for your experiment. Based on your application, the required measurement settings must be selected in step 2. For example, in the MA900 cell sorter, the user has a choice of 4 lasers and 12 fluorescents and 2 scatter parameters for which area, height, and width measurements are available. Once the measurement settings are selected, compensation tubes are automatically created in the software under the new experiment. Single stain compensation controls can be then run to create a compensation matrix. A multicolor sample, which will be sorted, is next run and analyze to create a gating hierarchy and define the populations that will be sorted. Lastly, the collection devices are set up and sorting is initiated. On the MA900 cell sorter, the user has a choice of sorting into 2- and 4-way tubes using Peltier-coupled tube holders that keep the sample cold at all times. Alternatively, single-cell sorting into plates can also be performed, again using special holders that provide a precise fit for different types of plates. So this was a complete overview of how to set up for sorting. We will next look into how to optimize sorting for different applications on the same instrument.
Hi, everyone. Thanks for being with us on this webinar. And thank you, Deena, for introducing the sorting workflow. My name is Miro, and I'm a Senior Field Applications Specialist with Sony Biotechnology. I will continue by looking in more detail at a few different applications user might encounter, all in the same day of sorting on an instrument. We'll cover yeast sorting and cleaning, followed by some high-parameter panel data, and finish up by looking at plate sorting of large cells. Let's start with yeast sorting. An example of this application might be working with these surface-displayed human antibody libraries for de novo discovery of therapeutic antibodies. Especially for a non-expert, the challenge is often to correctly identify targets using software tools, to maximize target yield, resolve clogs, and to leave the instrument clean for the users who run mammalian cells next. Very often, new users have a hard time identifying where the cell population is in the scatter gate. This is because each instrument software may look different and have different threshold and scatter default settings. However, with experience, you realize that, in fact, all flow cytometers detect cells using the same principles and should have the same 3 main settings. Let's briefly go through them. First, you want to adjust the forward and side scatter voltages to place the yeast population in view on the plot. You can also use either log or linear scales to visualize your data. In addition, I would make sure that the threshold is not set too high. Otherwise, half of your population may be ignored by the instrument. And if you're really not sure, you may run cell media alone to figure out where the debris ends and cell population would begin in your plot. Yeast cells typically have 2 subpopulations, the main population and a smear to the upper right-hand corner, which are most often aggregates. Because yeasts are a monoculture, there are no particular rules other than to gate on the majority of the population away from debris or very large and rare outliers. Further on, most users would like to formally exclude doublets using voltage pulse area versus pulse height. On the right, the doublets are shown on the second plot in the lower population within the red gate. It is commonplace to exclude doublets to achieve high purity. However, if the yeast target is very rare, contains unusual morphology due to budding, and requires enrichment sorting, then consider including those doublets. Let's look at fluorescent detection. Typical yeast applications do not require a high parameter number. On a laser-separated system with 12 detectors, it is easy to design a panel that requires no compensation. Also, Sony software contains a very useful manual compensation tool. Over or under compensation can be adjusted manually by dragging the populations in the plot, or you could follow the auto compensation wizard as well. Finally, 4-way sort capability allows sorting multiple populations to identify different binding affinity levels. You also want to take advantage of the software tools to gate on your target correctly. For example, you can create plot overlays of various control samples with your actively sorted sample. Finally, it's a good idea to adjust the scaling to see if any data are spread below 0. That way you're not missing a large number of your targets in your gating. After gating on your target, you've got to set a sort mode which will determine the levels of purity and recovery. One type of mode popular in yeast sorting is called an enrichment or yield mode. This mode sorts target cells even if the unwanted cells are present, and it can sort extra droplets to ensure cell recovery. For yeast sorting, this mode can help enrich for your target by hundreds-fold. Another big advantage is that it can be run extremely fast, close to your actual droplet rate. And if you have large sample volumes, 70-micron nozzle is the fastest nozzle to use. That way you can get through hundreds of millions of cells quickly to enrich for your target. Another mode type is called purity. Sorting in these mode types achieves a very high purity at the expense of aborting targets if a non-target is nearby, as shown here on the right. And for yeast sorting, this mode may be used after multiple rounds of enrichment sorting to clean up any remaining non-targets. Here's a quick example with some numbers. On the left is an example of a purity sort where cells were sorted slower and you lost about 10% of your targets to waste. However, the purity was very good at 98 or 99%. And on the right is an example of a yield sort where the event rate was 3 times higher and took only 45 minutes. The advantage there was efficiency and speed, and you've recovered close to 100% of your available targets. And even the purity in this case was not too bad at 96%. What are the challenges of sorting yeast? Well, very high concentrations of yeast cells are typically used for sorting. Therefore, sample line and nozzle clogging is often observed with this sample. Here on the left, you can see the event rate changed to 0, indicating a sample path clog, while on the right, the image shows that the droplet generation is disrupted because there is a clog in the nozzle. So it is especially important to watch out for clogs on the 70-micron nozzle, which is the narrowest of the 3 sizes that we offer, and therefore it is the most troublesome. So I would recommend that you refilter your cells throughout the sorting process, as often as every 20 minutes, because the onboard agitation may not prevent cell aggregation, especially if yeast are at a high concentration. Sony sorting chips are clog resistant, which is a huge benefit in yeast sorting. This is because the vast majority of the time, the clog occurs in the sample line or the chip's sample input needle. Critically, the droplet stream from the nozzle remains undisrupted and in calibration. On a Sony system, clogging can be resolved by sample line backflush with a click of a button. This process takes only 1 or 2 minutes and can even be done remotely. You may also want to refilter the sample before reloading it for continuing the sort. Once you've completed a yeast sort, it is time to clean the instrument to prepare it for the next user. If your instrument is used only for yeast, a simple bleach and water should suffice. However, in a multi-use scenario, it may help to completely isolate the sample path between yeast and mammalian samples. On Sony MA900 cell sorter, for example, it takes only a few minutes to switch from one sample line to another to prevent any chance of cross-contamination. For a very thorough cleaning of the entire fluidic system, you may use 1% bleach or 70% ethanol to maintain aseptic conditions. Sony MA900 sorters have automated cleaning protocols with customizable settings and even the cleaning reminders. So together, all these features give you the benefit of using the instrument, not only for yeast, but for a variety of other applications across different user groups. Now let's look at the kinds of topics one encounters when sorting immune cells with many subpopulations. Peripheral blood mononuclear cells, or PBMCs, are the classical example of a multicolor sample type. Although these cells are easy to work with, small and generally are clog-free, there's a wide variety of subpopulations with drastically different functions. Therefore, we typically identify the target cells with a lot of different markers of fluorochromes. Here you can see an example of a 12-color TBNK panel on a 3 or 4 laser MA900 instrument. Perhaps the question users ask is how many lasers and detectors does an instrument have, and what is the optical layout of the system? Here are the fluorochrome choices for MA900. This instrument has 4 lasers with 48 and 561 on the first beam spot, and the 405 and the 638 on the second. 48 laser is also used to excite the forward and the side scatter. As shown on the top, there are 5 detectors from the 48-561 laser line with yellow-green laser used to excite fluorescent proteins. And on the bottom, we have 7 detectors off of the 405-638 laser line. So to design your panel, you would pick one color from each column. If your samples do not express any fluorescent proteins, you do not need to use the 561 laser because you still get 5 detector channels from the 48 laser line. Shown in this slide is an example of the 12-color panel using just 3 lasers. Similarly, a 2-laser system shown in panel B on the right still gives you a capability to run a 12-color panel. So overall, MA900 optical design is flexible to support the transfer of 12-color panels from other instruments without changing optical configuration. Once you have your samples ready, it is pretty straightforward to create an experiment by selecting channels on the lasers, as Deena has described for you. If multiple fluorescent channels are activated, the software prompts you to follow the compensation wizard. So what are the best practices for multicolor flow cytometry? For one, you've got to have an unstained control without the viability dye in it. Single color controls can be prepared using antibody capture beads or with cells if your marker expression is sufficiently high. And they should be at a similar or higher brightness level to estimate the spillover values correctly. Once you have your controls, the compensation wizard will guide you through recording them one by one and calculating the compensation matrix, shown here in this slide on the right. Finally, please remember that all controls and samples are run at the same PMT voltage after calculating the compensation matrix. Changing detector sensitivity on the fly will invalidate that compensation math, and your gating will be affected. For those of you who might only be learning about compensation, I would pose a question. Why does compensation matter? Here is an example of the data with no compensation applied. Here we can see the telltale signs of multiple intermediate and diagonal populations, which biologically should be single positive. This obviously is a result of fluorochrome spilling into the adjacent detectors. After running single color controls and calculating the spillover correction values, we now observe only the true biological pattern of marker expression. Sorting the immune cells is fairly straightforward once you've gated on them. You would run a sort sample at 1/5 the frequency of the nozzle. So for example, on the 70 micron nozzle, you would run at 12,000 events per second. Then you would probably sort 4-way into fax tubes with media where each stream has been accurately targeted. You would also observe the event rate and sort efficiency during sorting and troubleshoot any encountered clogs, as I've discussed on previous slides. Purity check sacrifices some sorted cells to give us the confidence that the sort went well. Here are some of the critical factors you should consider. You should be concerned about the carryover in the sample line, and I would suggest running sample line rinse before the purity check. Also be aware that cells may fall out of the sort gate, which results from the dye washing off, photobleaching, or normal detector variability. You may also want to look if scatter shifts as an indication of viability post-sort, or restain cells with a viability dye. Overall, I'd say that a purity of more than 98% and efficiency of over 80 or 90% can be expected for experiments that are set up using best practices. For my third topic, I will discuss sorting large cells into a plate, which is yet another application that Sony MA900 excels at. When we say large cells, we often mean mammalian or even plant cells over 20 microns in diameter, such as cancer cell lines, primary fibroblasts, stromal cells, cardiomyocytes, epithelial cells, and neural cells. We also know that many users require sorting these cells into a plate for cell cloning or single-cell PCR applications. You might have guessed correctly that sorting large cells is challenging due to their size and sensitivity to shear stress. This is why sample preparation and nozzle choice, sort settings, and plate hardware are all so critical to this process. For those of you who are interested to learn a bit more about this application, I would suggest looking at my previous webinar recorded in May of this year. Today, I will emphasize some of those best practices, as well as discuss additional useful features you might need for plating large cells. Here's one good example, which is U2OS cell sorting with index function. This slide shows a typical gating scheme, starting from cells, most of which are viable, followed by singlet gating, and sorting for different levels of GFP expression at one cell per well. The two plots on the right also show you an index data analysis feature. You might ask, what is index data? Index data refers to each cell's fluorescence and scatter data correlated with plate's well position. And it is most useful when doing single-cell deposition into multi-well plates. As you recall, we started our sorting day on Sony MA900 with yeast cells and a 70-micron nozzle, followed by sample line exchange and system-wide bleach cleaning. And then we sorted PBMCs into a tube. This slide here describes how one can easily switch from tube sort to a plate sort format, only in a couple of minutes. Once the plate hardware is set, it is time to filter the cells and then preview and gate on live cells, making sure that your forward and side scatter detector voltages are set low enough not to miss any cells. One tip I can give is to start with a very low forward scatter setting and incrementally increase it until you see the main population outline. Another tip is to do a quick sort of the cell gate onto a glass slide to make sure that the cells are there instead of debris. Finally, for single-cell cloning, you must gate out doublets. Now let's go over the instrument setup. From my webinar in May, you might recall that large fragile cells should be sorted using a 130-micron nozzle. This low-pressure nozzle gives the best cell viability and the most precise droplet targeting into a well. Further, you must use a single-cell mode to prevent two targets from getting sorted into one well. It is critical to realize that just because you've gated on singlets, it does not mean that two singlets cannot be in the same or adjacent droplet. I would also suggest keeping samples chilled and agitating, and run sticky cells more dilute at a lower event rate. Most often, you only need 96 targets, and if they are above the 1% in abundance, then you can run them at under 200 events per second, and your plate will be done in under a few minutes. Finally, I suggest sorting cells into warm condition media for best cell survival. Sony sorters provide users with a variety of plate holders to fit almost any plate type. Critically, to reduce the chance of depositing a droplet on the sidewall of a PCR well or a 3D4 tissue culture well, I would suggest using an angled plate holder. As shown in the middle of the slide, this angle of the plate holder helps the droplet to reach the bottom of the well, where the lysis buffer is. This is how we can achieve a very high plate well occupancy rate of about 95%. To do so, you must leave the plate cover on and then align the droplets to the well center, which we will look at next. Sony sorters auto-calibrate stream angles to hit the centers of fax collection tubes. However, as shown on the right, additional alignment tools are available for fine-tuning the plate coordinates. Importantly, you can perform a test sort onto a plate cover using your cells in the droplets, and you can adjust each corner's alignment independently of the other corners. It takes only a couple of minutes to follow the steps outlined here, and a lot of times 96-well plates are already close to perfect alignment. So if you're sorting large cells, 130-micron nozzle will help to deliver them to the centers of the wells. At the top of the slide here, you can see a puddle of 10 to 20 droplets that land perfectly. On the other hand, on the bottom is an example of using a smaller diameter nozzle, which may not get your cells into the well. Sony software allows you to sort different populations and different cell numbers in each well. So after you've performed plate alignment, you would then create a plate sort layout for your targets and begin sorting. Let's go back to the index sorting and look at how does it work. If a cell satisfies a sorting criteria, the instrument will place the droplet with that cell in it into the well. There is 1 cell, 2 cells, 3 cells, so on and so forth. As the previous slides showed, the MA900 software records indexed single-cell data to correlate fluorescence data with each target's well position. In this slide on the right are a few examples of our data analysis tools. One is called Well Select Mode, which shows each selected well in the plots. Another mode is Gate Select Mode, which identifies all wells with targets from a particular gate. These software features allow us to perform a metadata analysis for downstream cloning and sequencing experiments. Here is an example of what you can do with index data. Because the data are exportable as a CSV file, you may correlate your gene expression level with the fluorescent marker expression, as shown here in the plot on the right. So, in summary, the main message of our webinar is that there are many challenging applications in cell sorting. The learning curve can be steep, and there are a number of key sort parameters for you to consider. Finally, it is fair to say that it is hard to design a versatile, automated system with a variety of features to cover all of the applications. And we hope that after today, you will have a better idea on how Sony MA900 streamlines and automates the sorting workflow. Thank you so much for your time, and now we can take any questions.
Thank you, Dr. Soni and Dr. Koulnis, for your informative presentation. We have 2 more questions for our audience. If you could take a moment to look at the questions and participate, we greatly appreciate it. Thank you for your participation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. Our first question is, does the Sony sorting chip get saturated after running one experiment? What if I want to use the chip for the entire day for sorting?
I can take that question. So the Sony sorting chip can be scanned and used for the whole day for 24 hours. It does not get saturated, regardless of how many samples you run. If there is any air or cells that get trapped or clogged in the chip over the course of the day, those can be resolved simply by priming the chip or doing a back flush as we showed in our presentation.
Thank you, Dr. Soni. The second question is, if yeast cells are subjected to successive rounds of enrichment sorting for faster throughput, what is the impact of this on their viability?
I can take that. So generally, the yeast cells are very hardy, and you can sort them at a high rate, and their viability doesn't suffer. I mean, I have customers who sometimes tell me that they sort these cells into a dry tube because they survive so well. So I would say that the impact can be minimal, especially if you allow cells to recover after the first round of enrichment sorting. So you grow them for a couple of days, then you sort them again. So overall, the impact on viability is usually minimal.
Thank you, Dr. Koulnis. Our next question: is there any advantage to using single stained cells versus single stained beads for compensation?
So I can also take that. You can definitely use either cells or beads for the compensation. So the advantages of the, I will answer it this way. So the advantages of using the compensation beads is that if you don't have a good, strong cell positive control, the bead provides a high level of fluorescence capture on the bead surface to have a good positive control. However, the advantage of using cells is that you estimate the autofluorescent background best. A lot of it kind of does depend on the setup of your experiment too, which fluorochromes you're using. But generally, I stick to either using all beads or all cells, and I realize there's a lot more to that, and it does become kind of situation dependent. But generally, you can use either one as long as your single positives are nice and bright.
Okay, thank you. We have another question. I am using very large cells with elliptical shape for sorting. Any tips on how I can improve single cell deposition on 96 well plates?
Yeah, I can answer that as well. So we've already covered a lot of those aspects, but I can kind of restate them again. So when you sort cells into a plate, it's always good to use a 130 micron nozzle, especially if the cell is very large and irregularly shaped. So certainly 130 nozzle helps, as well as using 3-drop mode. So there are sort modes that deviate for each target cell, either one droplet, so the cell is in that droplet, or you could also set up a cell sorter to sort using a 3-drop mode. Depending on on the instrument you're using, 130 micron nozzles, for example, sometimes they can by default sort 3 droplets at a time. So definitely use a 3-drop mode and make sure that you align the plate really well using cells when you're doing plate alignment, when you're doing test sort onto a plate cover. I would also say make sure that you use enough buffer in your wells, so it's not, it has the largest surface area to basically capture that droplet or the 3 droplets if you're using. Last tip would be to perhaps not sort too fast because the more targets, if you're doing multiple targets per well, the more targets you have, the more staining you will experience. And I guess perhaps that's a different part to this question because we're talking about single cell deposition. But in any case, if you slow down the total rate, that reduces the staining and improves the targeting into a well.
Thank you, Dr. Koulnis. We have another audience question. What about index sorting software?
Yeah, so I've seen this question come through. I think we've covered pretty extensively in the last 5 slides of our webinar. So I would just guide that audience member back to those slides after this has been made available for the post-recording view.
Okay, great. We have another question. Do the sorting chips have a limited shelf life and cannot be used when scanned after a period of time?
Yeah, so I can take that question. Sorry, Miro.
Go ahead.
All right, thank you. So like we discussed before, once the sorting chip has been scanned, it has a 24-hour lifetime for usage. Of course, if we talk about shelf life per se, which means just storage at the bench, there is no expiration date. You purchase the chip, you can pretty much use it for, you can store it for years to come.
Okay, thank you, Dr. Soni. Our next question, how does the Sony MA900 address biosafety level when sorting human cell or cell lines? Would these cells be categorized as BSL2 or BSL2 plus?
Yeah, I can take that one as well. So the Sony MA900 and all the other Sony sorters as well, they have a provision where they can either be installed inside a biosafety cabinet, which is a custom product available from Sony that enables our customers to sort BSL2 and as well as BSL3 type of applications. We also have an ability to attach the cell sorter to an aerosol management system. And then depending on the kind of cells you're using, really human cells, and if there are any viruses in it, it would get classified either as BSL2 or BSL2 plus. I believe there is a classification chart available from the NIH that could guide us a little bit better on that. But other than that, there are several features on the MA that make it a fairly, airtight system, if you will, in terms of aerosol containment itself.
Thank you, Dr. Soni. We have another question. This is the last audience question so far. This is a question about how to improve a better cell sorting test before real cell separation.
I think I can answer that. If you're asking about tube or actually, in fact, for both tube or plate sorting, if you are talking about targeting of cells into those collection devices, you can do a quick test sort. It really doesn't have to take a lot of cells because each droplet with a cell in it is only around 3 to 10 nanoliters. You can do a quick test sort onto a parafilm, covering the tubes or a plate cover, making sure that the droplets will hit the centers of those wells, centers of those tubes. Like I said, it doesn't take many cells to do this test. To get a small puddle, you need only 30, 40 cells. So you would do this test sort, and that will make sure that the cells can go down the tube or into the plate.
Thank you again, Dr. Soni and Dr. Koulnis. Do you have any final comments for our audience?
Well, if there are any more questions, please feel free to get in touch with us at sonybiotechnology.com.
Great, thank you so much. And before we go, I would like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. We would like to once again thank Dr. Soni and Dr. Koulnis for their time today and for their important research. We would also like to thank LabRoots and our sponsor, Sony Biotechnology, for underwriting today's educational webcast. You can view the webinar on demand. LabRoots will alert you via email when it's available for replay. That's all for now. Until next time, goodbye.